Hello everyone, this is UPSC 2023 Agriculture Optional Question Paper. So the question is enlist various photosynthetic pigments, give a detailed account of chlorophyll synthesis along with the enzymes involved in the process and also mention the role of the carotenoids in the photosynthesis. So basically there are three parts in this question. First of all, we have to enlist the photosynthetic pigments and then chlorophyll synthesis pathway along with the enzymes involved in the process and then role of the carotenoids in the photosynthesis. So this is the brief introduction. You can write in your own way about in the introduction you can tell everything about like photosynthetic pigments also, whatever you want. And then this is the main body part, photosynthetic pigments. So basically there are three types of photosynthetic pigments: chlorophyll A and B and carotenoids. These carotenoids are fat soluble yellow to orange color pigments, and these are classified into two types: carotenes and xanthophylls. The examples of carotenes are alpha and the beta carotene, and xanthophylls examples are lutein, zeaxanthin, etc. Then phycobilins, these are found in the cyanobacteria or algae. Example is phycocyanins. So these are the photosynthetic pigments which are responsible for the photosynthesis process. Now the role of the carotenoids in the photosynthesis, basically the major role of the carotenoids is there are two major roles of carotenoids. First of all, they act as a photoprotective agents, that is preventing the harmful photodynamic reaction. So they dissipates the excess absorbed light energy as a heat. Now they act as a accessory light harvesting pigments, extending the spectral range over which the light derives photosynthesis. So basically carotenoids absorb the light in the blue to green region, that is between 400 to 500 nanometer, where chlorophyll absorption is weak. So that's why the region in which the chlorophyll can't absorb the light, carotenoids can absorb the light in that region. That's why they act as a accessory light harvesting pigments, extending the spectral range, that is between to 400 to 500 nanometer, over which the light derives photosynthesis. Then they stabilize the thylakoid membranes. Thylakoid membranes, they are found in the chloroplast, so ensuring the proper assembly and the orientation of pigment protein complexes for the efficient electron transport. So these are the roles of the carotenoids in the photosynthesis. Now this is the chlorophyll synthesis pathway. So first of all, glutamate, that is found in the chloroplast, it is converted to the 5-amino levulinic acid in the presence of the enzyme glutamyl tRNA reductase. Then this ALA is converted to the protoporphyrin 9, and these three enzymes are involved. These are the three intermediate steps through which जो है this ALA is converted to the protoporphyrin 9. So the enzymes are uroporphyrinogen decarboxylase, coproporphyrinogen oxidase, protoporphyrinogen oxidase. Now this protoporphyrin 9 will convert to the protoheme 9 under the presence of ferrochelatase, and then it will convert to the heme pigment. But in case of plants, when magnesium enters in the presence of ATP and the enzyme is Mg chelatase, it converts to the Mg protoporphyrin 9. Then it is converted to the Mg protoporphyrin 9 monomethyl ester in the presence of the enzyme Mg protoporphyrin 9 methyl transferase. Then it is converted to the DV, that is divinyl protochlorophyllite, in the presence of Mg protoporphyrin 9 monomethyl ester cyclase enzyme. Then this DV protochlorophyllite is converted to the monovinyl protochlorophyllite in the presence of divinyl reductase enzyme. And then this MV protochlorophyllite is converted to chlorophyllite in the presence of protochlorophyllite oxidoreductase. Then this chlorophyllite is converted to the chlorophyll A in the presence of chlorophyll synthase, and this chlorophyll A converts to chlorophyll B in the presence of chlorophyll A oxygenase enzyme. So this is the pathway that from glutamate present in the chloroplast, it converts to the ALA in the presence of these three enzymes, then protoporphyrin 9. Then when Mg enters, it converts to Mg protoporphyrin 9, then monomethyl ester, then divinyl protochlorophyllite, then MB, then chlorophyllite, then chlorophyll A and chlorophyll B. So in this way you can attempt your question. Thank you and keep watching my videos.